Have you ever sat in a doctor's office, a small needle pricking your arm watched as your blood filled a little tube? You probably put a cotton ball on it, got a colorful bandage, and went on with your day. But have you ever stopped to wonder what happens next? Where does that little vial of you go? It embarks on a fascinating journey behind the closed doors of a clinical laboratory. This hidden world of science and technology springs into action to unlock the secrets in that single drop. It's far more intricate than most imagine, a critical first step in your health story. That small tube, labeled with your name and a unique barcode, is like a message in a bottle. It's placed into a secure transport bag often kept at a specific temperature. It travels across campus or to a specialized lab miles away, joining hundreds of other samples. The journey is meticulously tracked. The barcode is the key to prevent mix-ups. From the moment it leaves your side, professionals guard its safety and accuracy. At the lab, the real work begins. A technician scans the barcode and logs the sample into the lab information system. That digital footprint links the tube to your electronic health record, creating an unbroken chain of custody. Not all tests are created equal, and your body offers many different kinds of clues. The most common type of test, and the one we are most familiar with, is the blood test. Blood is a rich source of information, acting as the body's transportation highway. It carries oxygen and nutrients, it carries hormones, it carries waste products. By analyzing your blood, scientists can check for signs of infection, measure your cholesterol, measure your blood sugar levels, assess how well your liver is working, assess how well your kidneys are working, and even look for markers that might indicate certain diseases. For a blood test, this often involves a centrifuge. Picture a fast-spinning carousel. Vials of blood are loaded into the rotor, the centrifuge whirls and separates the blood. Red blood cells sink to the bottom. White blood cells and platelets form a middle layer. Plasma or serum rises to the top. Different tests need different layers. Many chemistry tests use serum or plasma. The technician selects the correct part and extracts it with a pipette. For microbiology, a tiny smear is placed on a glass slide and stained for microscope review, or the sample is spread onto a petri dish with nutrient agar to culture organisms. For genetic tests, DNA is extracted from cells. Reagents break open the cells. Each protocol is a precise recipe for that test. This meticulous prep is quality control. If preparation is wrong, results can be incorrect. That's why lab pros are highly trained and follow strict protocols to ensure accurate, interference-free results. Modern medical labs are filled with amazing technology. These are the high-tech tools that do the actual work of analyzing your samples. One of the workhorses of the chemistry lab is the spectrometer. The spectrometer shines a beam of light through your prepared sample, like serum from your blood. Different substances absorb different amounts of light. Different substances absorb different wavelengths of light. The spectrometer measures how much light passes through. It also measures how much light is absorbed. By analyzing the light pattern, it calculates precise concentrations. It measures things like blood sugar. It measures things like liver enzymes, fast and accurate. For looking at cells and microorganisms, the classic tool is the microscope. Today's lab microscopes are linked to cameras and computers. They can magnify objects thousands of times. A lab professional can manually count and examine different types of blood cells. They look for abnormalities in cell shape. They look for abnormalities in cell size. When it comes to genetic testing and infectious disease detection, the PCR machine is revolutionary. PCR stands for polymerase chain reaction. Think of it as a molecular photocopier. If there's a tiny amount of DNA or RNA in your sample, PCR can amplify it. It can make millions of copies. It can even make billions of copies in just a few hours. That amplification makes the genetic material much easier to detect. It's sensitive enough to find the smallest trace of an infection. It can also identify specific genetic sequences. Many analyzers are fully automated, running hundreds of samples an hour with minimal human intervention. Once the machines have finished their analysis, they produce a stream of raw data, numbers, graphs, and images. But this data is meaningless on its own. The next crucial step is interpretation. The lab's LIS takes the raw data and compares it to reference ranges. A reference range is the set of values considered normal. These ranges are established by testing many healthy people adjusted for age, adjusted for sex. What's normal for a child may not be normal for an adult. Results outside the range are flagged too high or too low. This is the first indication something may need attention. However, abnormal doesn't always mean disease. Many things affect results. What you ate, time of day the sample was taken, or even your stress level. Laboratory scientists review results, pathologists review results. They may re-examine the sample or check machine performance. After verification, final results are compiled into a clear report. Every single day, before any patient tests are run, technicians run control samples. These are commercially prepared samples with known values. For example, they will run a control serum where the manufacturer has determined glucose is exactly 100 milligrams per deciliter. The machine must produce a result that is extremely close to that known value. Labs also participate in proficiency testing several times a year. An external agency sends the lab blind samples. The lab doesn't know what the results are supposed to be. The agency then compares the lab's results to those from hundreds of other labs around the country. This is like a final exam, proving that the lab can consistently produce accurate results. The clinical laboratory is a field in constant evolution, and the changes we are seeing today are nothing short of revolutionary. One of the biggest drivers of this change is automation. In large modern labs, you'll see entire tracks and robotic arms moving racks of samples from one station to another. A sample can be logged in centrifuge, divided into smaller portions, and sent to multiple different analyzers without ever being touched by a human hand. This total lab automation drastically increases speed and throughput, allowing labs to process thousands of samples per day. Beyond just moving samples around, artificial intelligence, AI, is beginning to play a powerful role in interpreting the data. For example, AI algorithms are being developed to analyze images from microscopes. An AI can be trained to scan a digital image of a blood smear and identify abnormal cells with a speed and accuracy that can sometimes surpass a human expert. Despite these incredible advances, challenges remain. The risk of sample contamination, though minimized by automation and strict protocols, can never be completely eliminated. Human error, while reduced, is still a factor. A sample collected improperly before it even reaches the lab, or a simple data entry mistake can have downstream consequences. That is why the focus on rigorous training, double-checking critical steps, and fostering a culture of quality and safety remains paramount, even in the most technologically advanced labs. We are seeing the rise of point-of-care testing where simple tests can be done right in the doctor's office or even at home, giving immediate results. Technology is also enabling the development of tests that require smaller and smaller sample volumes, like a single drop of blood. The core mission remains the same: to provide fast, accurate, and reliable information that empowers doctors and patients. The lab is, and will continue to be, the silent partner in your healthcare, constantly innovating to help you live a healthier life.